Welcome to another episode from the Veterinary Medicine Digest. Today we are embarking on a comprehensive exploration of a vital yet complex area of neuropathology, the detection and characterization of axonal injury in the central nervous system, with a specific focus on the Ovine model. For those of you who have spent time in the diagnostic laboratory or in forensic research, you know that the ability to accurately time an injury and assess the extent of neural damage is often the cornerstone of a successful investigation. At the heart of this pursuit is a remarkable molecule known as Beta Amyloid Precursor Protein. To understand the current retrospective research into sheep encephalopathies, we must first build a foundation in the basic biology of our primary marker. Beta amyloid precursor protein is a ubiquitous transmembrane glycoprotein produced within the neuronal cell body. Under normal physiological conditions, it plays an essential role in cell adhesion, synaptic modification, and endogenous neuroprotection. What makes this protein so valuable to the pathologist is its mode of travel. It is moved through the axon by fast anterograde axoplasmic transport. Imagine the axon as a highly efficient highway. As long as the infrastructure is intact, the protein flows smoothly to its destination. However, when a traumatic or toxic insult occurs, this highway is disrupted. The mechanical shearing or compressive forces of a head injury or the biochemical assault of a neurotoxin leads to a distortion of the axonal cytoskeleton. This physical damage triggers a cascade of events. Microtubules disassemble, and the influx of calcium activates various deleterious enzymes that further break down the structural integrity of the nerve fiber. As the fast axonal transport system fails, the beta amyloid precursor protein begins to accumulate rapidly at the site of the injury. It is this accumulation that we can detect using immunohistochemistry, essentially using the protein's own travelogue to pinpoint exactly where and when the damage occurred. Historically, in the context of human forensic pathology, the earliest reported time at which axonal injury could be detected using beta amyloid precursor protein was 35 minutes. However, recent retrospective studies involving road traffic collisions have pushed this window even earlier. By examining patients who died nearly instantaneously or within a few minutes of a collision, researchers demonstrated that immunoreactivity could be seen in less than 30 minutes. This suggests that the process is incredibly rapid, especially when the magnitude of the force is high, such as in high-speed vehicle accidents or severe impact injuries. While human studies provide the temporal framework, the veterinary world offers unique models to study these mechanisms in depth, particularly through the lens of clostridial diseases in sheep. One of the most significant enteric pathogens in small ruminants is Clostridium perfringens type D. This bacterium produces a potent neurotoxin known as the Epsilon Toxin. The journey of this toxin is a fascinating and terrifying example of biological warfare. It is produced in the intestine as a relatively inactive prototoxin and then activated by intestinal enzymes like trypsin. When certain conditions favor the rapid proliferation of these bacteria, such as a sudden intake of starch-rich feed, often called overeating disease, large amounts of the toxin enter the systemic circulation. The Epsilon toxin is a vascular-centric neurotoxin. It binds to microvascular endothelial receptors, particularly those forming the blood-brain barrier. This binding leads to increased vascular permeability, allowing an abundant extravasation of plasma proteins and fluid into the brain parenchyma. This results in two distinct syndromes based on the dose and duration of exposure. In the acute form, where high levels of toxin reach the brain, the animal suffers from severe generalized cerebral edema, leading to a rapid rise in intracranial pressure and often sudden death. However, our deep dive today focuses on the protracted neurologic syndrome, which occurs when lower doses of the toxin are absorbed or when the animal is partially immune. This form of the disease is characterized by the development of bilaterally symmetrical necrotic lesions in specific regions of the brain, a condition pathonomically known as focal symmetrical encephalomalacia. These necrotic foci are typically found in the basal ganglia, internal capsule, thalamus, and cerebellar peduncles. It is within these lesions, and particularly at their periphery, that we find a treasure trove of information regarding axonal injury. In the necrotic areas of focal symmetrical encephalomalacia, beta amyloid precursor protein is overexpressed, highlighting injured axons that have developed into swellings known as axonal spheroids. These spheroids represent the morphological manifestation of disrupted axonal transport. As the protein accumulates, these swellings become visible under light microscopy, but here is where the diagnostic challenge begins. Axonal injury is a heterogeneous process. Not every injured axon looks the same, and not every marker detects every type of injury with the same efficacy. A critical recent study sought to compare the effectiveness of a panel of antibodies in detecting the complete range of axonal injury in sheep with focal symmetrical encephalomalacia. The researchers evaluated a battery of immunohistochemical markers including beta amyloid precursor protein, 3 subunits of neurofilament protein, protein gene product 9.5, ubiquitin, and synaptophysin. This comparison is vital because the rate at which these different molecules are transported along the axon varies significantly. Vesicular molecules like beta amyloid precursor protein and synaptophysin move quickly, while cytoskeletal proteins like neurofilament protein and cytosolic markers like ubiquitin move more slowly. The results of this study confirm that beta amyloid precursor protein remains the most sensitive and early early marker of axonal injury, capable of detecting damage within minutes of the insult, it detected significantly more spheroids, both large and small, and produced a strong, easily visualized signal compared to the other markers. However, the researchers also found a complex pattern of labeling. Different subsets of spheroids were often labeled by different antibodies, suggesting that they were at different stages of temporal evolution or possessed different protein compositions. For instance, neurofilament protein labeling varied according to the specific subunit examined. The 68 kilodalton and 200 kilodalton subunits tended to detect larger spheroids, while the 160 kilodalton subunit labeled many smaller ones as well. Ubiquitin immunoreactivity, which suggests that structures within the reactive swellings are targeted for degradation, detected fewer spheroids and yielded a weaker signal. Synaptophysin, despite its fast transport rate, showed very scant labeling of spheroids in this model. This heterogeneity is a crucial takeaway for the diagnostician. Using only a single marker might lead to a significant underappreciation of the total amount of axonal damage present in a lesion. Building upon this, another layer of complexity arises from the technical aspects of immunohistochemistry itself. When we use antibodies to detect beta amyloid precursor protein, we must be aware that they can be directed against different epitopes or antigenic determinants on the molecule. There are three primary domains of the protein: a large extracellular N-terminal domain, a transmembrane domain, and a short cytoplasmic C-terminal tail. A recent optimization study in sheep brains compared 4 different antibodies: 22C11, Y188, NAB228, and C1/6.1. While all 4 antibodies were capable of detecting axonal spheroids in focal symmetrical encephalomalacia lesions and traumatic brain injury models with similar efficacy, they behaved very differently regarding background staining. The Y188 antibody, which is highly prized in some rodent studies for its specificity, produced robust and diffuse labeling of uninjured axons in sheep, which was later determined to be a specific reaction with myelin sheaths. This is an important cautionary note for veterinary pathologists. If an antibody produces strong background staining of normal myelin, it becomes much more difficult to visualize and quantify small axonal spheroids. By contrast, the 22C11, NAB228, and C1/6.1 antibodies provided a clear signal with minimal background, making them more suitable for diagnostic work in the ovine brain. This brings us to the broader biological context of our marker. While we primarily use beta amyloid precursor protein to detect injury, its normal distribution is still a subject of ongoing research. Recent studies using highly specific antibodies have clarified that, in the central nervous system, this protein is specifically expressed in neurons. It is not detected in major types of glial cells such as astrocytes, microglia, or oligodendrocytes, even under neuroinflammatory conditions or following traumatic brain injury. In neurons, it is primarily localized to the Golgi complex and late endosomes, which aligns with its role in the secretory pathway. The stability of the soluble forms of this protein is another area of interest. When the protein is cleaved by secretases, it generates soluble derivatives that are secreted from the cell. Research has shown that these soluble fragments are remarkably stable in the central nervous system and do not undergo further rapid cleavage even under conditions of trophic factor deprivation. This stability might be important for their hypothesized roles in neuroprotection and synaptic signaling. Furthermore, an interesting interaction has been identified between the protein and a chaperone protein in the endoplasmic reticulum known as glucose-regulated protein 78 kilodaltons. This interaction likely facilitates the correct folding and trafficking of the protein, and its disruption could have implications for disease states. In the case of more advanced focal symmetrical encephalomalacia lesions where lytic necrosis occurs, the pathology takes an interesting turn. Macrophages that invade the necrotic area are found to have phagocytosed not only myelin debris but also axonal fragments that are immunopositive for beta amyloid precursor protein and neurofilament protein. This engulfment of damaged axonal material by macrophages is a notable finding that helps us understand how the brain attempts to clear the debris of a focal insult. So, why does the brain develop these specific, symmetrical lesions in focal symmetrical encephalomalacia? This phenomenon is known as selective vulnerability. Most neurologic diseases deliver an insult to the entire brain, in this case, via the systemic circulation, yet the damage is often confined to specific neuroanatomic regions. While the exact reason for this in the sheep model remains elusive, it likely reflects a higher susceptibility of the neural parenchyma or the local vasculature in those specific areas to the epsilon toxin. This concept is seen in other conditions too, such as carbon monoxide poisoning in humans, which targets iron-rich areas like the globus pallidus. Understanding the nuances of these lesions is not just an academic exercise, it has significant diagnostic specificity. While necrosis, or malacia, is a common endpoint for many insults, including vitamin deficiencies or ischemic events, the specific neuroanatomic pattern of distribution in focal symmetrical encephalomalacia is considered diagnostic for Type D neurotoxicity. By using a robust panel of markers for axonal injury, pathologists can more accurately characterize the stage and severity of the disease, even when traditional markers might be faint or incomplete. As we conclude our deep dive, it is clear that the study of axonal injury in sheep encephalopathies is a field defined by both its technical challenges and its diagnostic rewards. The evolution of beta amyloid precursor protein as our primary marker has revolutionized our ability to detect damage within minutes of an insult, providing a critical window into the rapid pathophysiology of both trauma and toxemia. By carefully selecting our antibodies, understanding the timing of protein transport, and recognizing the heterogeneous nature of axonal spheroids, we can better decode the messages written in the injured brain. Thank you for listening. The titles for the reference papers are in the video description. From Vet Med Digest, where we keep minds inspired, hearts light, and tails wagging.